Well, good afternoon, everybody. Can you hear me? Yes. Okay, better. Okay, great. Well, so good afternoon. It is my pleasure to introduce today's speaker, Dr. Paul Esker. It is on.
It's on.
It is on.
Can hear you.
Can you hear me now? So, good. Okay. Yeah, better. All right. It is my pleasure to introduce today's seminar speaker, Dr. Paul Esker from Penn State. If probably you don't know him, he's an expert in plant disease epidemiology and field crops plant pathology. Dr. Esker's extensive academic and research background spans multiple institutions, even countries. We were just talking about Costa Rica a few minutes ago. And if you see his background, he's very extensive. He's going to be talking about machine learning, one of the hot topics nowadays in multiple areas, actually. And so perhaps you have been following the news. This year's laureates in the Nobel Prize in physics were working in machine learning. And so the idea here is to learn from Dr. Esker about his expertise, how he's leveraging that knowledge in AI with increasing yields and things that are happening in agriculture. Dr. Esker earned his bachelor's in genetics and bacteriology from the University of Wisconsin-Madison, and his master's and PhD in plant pathology and statistics from Iowa State University under the late Dr. Forrest Nutter, a renowned expert in quantitative epidemiology and phytopathometry. And after completing an NSF postdoctoral fellowship in biological informatics, he held faculty employment at the University of UW-Madison and the University of Costa Rica before joining Penn State. And currently at Penn State, he leads the Fields and Forage Crops Extension Program and co-chairs the International Agriculture and Development Dual Degree Program. His innovative research focuses on using advanced modeling techniques to study plant disease epidemiology and integrated pest management. And without further ado, Dr. Esker, thanks much for coming. Let's help me joining Dr. Esker here.
So can everybody hear me all right with this one? Okay, I'm going to use it. I'm going to try not to wander too much, but these three screens are quite impressive, actually. So it is an honor to be here with everybody. And Christian, thank you for the invitation. I really want to especially thank all the staff and everything with the logistics. I mean, to get all the information in July already for something in October is kind of unusual in an academic culture, at least in Penn State, in that respect. So yeah, I'm going to talk about, I used this title actually with an advisory committee to the dean in April, and somebody really liked it, so I kept it. But I'm always curious if I shouldn't say protecting crop yield more and how we look at it at the end of the day, because we're looking at the factors that drive it and how to build scenarios. But that being said, it's how do we take observational data and put something tangible to it in this respect. So my program, I've been there since 2017. This is my current staff up there. I have two postdocs, and Tyler runs my field program, and Miranda is actually a data scientist in my unit. That's one of the keys to run a program like this today is really broad sense. Her background's more in microbiomes and bioinformatics. So she builds a lot of our data pipelines. And the last part of the talk, she's really been instrumental in what we do in our current iteration with some of our data platforms. Current graduate students, I've talked to the students, they all do something slightly different. There's Laundry, the first one who does wheat, Evan works on hemp, Savannah works on soybean, and so it's fusarium, a mix, and Pretilicus, so nematodes at that level. And I have a new student who actually works for our Department of Agriculture and Diagnostics, who will be joining in January. We have a strong support staff. Flavia, just mentioning her, she's from Brazil. She's a visiting scholar, very similar to a lot of the students I've interacted with while I'm here in that respect. And since I've been back, we've been able to place our postdocs in 2 faculty positions, and one's a data scientist for Syngenta in Switzerland. So I think what we're doing is working all right, and we've finished off. I had 3 students graduate this year in different roles. And all of them have had programs that they're interested in teaching. We build their program around that. If they're interested in extension, we work that way. I do unofficially have a 3-way split. If I go into our system, it doesn't actually show up as such, but I teach, I do extension, and I do research. I don't know what the weights mean anymore at this point, nor do they really matter, so I'm okay with that. Just briefly, we run a very mixed program. This just illustrates it from what we do maybe in a wet lab setting. So this is some of the work for nematodes that Savannah will be working on. We have a strong field aspect. Lonry, actually part of his research is done in Ethiopia with CIMID in the Ethiopian Institute of Agricultural Research. Mariah, who just started her PhD at North Carolina State, did her master's and secured an NSF graduate research fellowship, is actually working on nematodes as well. Moved back home to work with Adrian Gorney. We do use technology where it's appropriate in that system, but in our case, and a lot of what we do is work more from a contract side. I find the investment in the equipment up front is maybe a limiting factor in what we can do from working with some of the sector that way. For those who aren't familiar, you see New York to the north of us, and then in Pennsylvania. One of the advantages I think we have for epidemiology is the topography drives a lot of microclimate variation. The way the Appalachians run across it, it gives us unique areas that can align to other areas in that respect. So a lot of what we've been doing is trying to get a grasp of some of the pathogen diversity in some of that aspect. This global plant health assessment is a large project that we secured funding from OECD, and we work with about 90 to 100 researchers around the globe, looking at the impact of plant diseases on ecosystem services. That's an ebook available at the International Society of Plant Pathology if you're interested. And we're actually in a phase 2 right now. So there, if you'd like to hear more, you can always reach out to me to ask about that in some detail. My teaching and / administration, as Christian said, I am the co-chair and kind of the professor in charge of our International Agriculture and Development. This is a dual degree graduate program at the master and PhD level. We have students from 13 graduate programs across 2 or I think it is 3 colleges and 1 inter-college graduate program in ecology. And so the students do their coursework and research in their home department. And often what INTAD focuses on is the social science aspect of what they may do. And so their coursework is around that. I co-teach the capstone seminar course in INTAD 820 with a rural sociologist, for example, in that respect. I also teach graduate epidemiology, which is why I say I have a 3-way split, because it was on the contract, even though it doesn't show up in the system in that respect. Some of the things I have to do in that role, we were a group of us were at Coffee in Costa Rica in June, kind of talking about some of the collaboration. The other aspects, this is an example, this is also freely available. Through that course, we've written chapters that ended up in this book for an undergraduate course. And this is an ebook on Everyone Needs to Eat and Understanding Food Security and Global Agriculture. So this is how we take it from the classroom into something practical for other things. I also have an extension appointment. Some of you may know Alyssa Collins. So in our system, I'm lucky because none of us, our extension appointments are very small. And they've shrunk in some degree. I think the top end appointment might be 60%, mine is 25. Alyssa is our director of our Southeast Research Farm in Lancaster County. She and I collaborate in a lot of the extension, and that's how we make the engine work. A lot of conversation about tar spot. We also have it, so it's been a good conversation. Evan Buckner is one of my graduate students who was at the University of Arkansas. He's working on hemp, and it's always fun to have those field days because it's a very eclectic group of people. So I'm not actually sure what the gentleman in the blue shirt's doing there, to be honest. Probably having a good sniff of something. We are funded broadly by the commodities, by the federal government, and internally, you'll see the last part more about that at the end. And so we've had success with our Northeast Sustainable Agriculture Research and Education Program. They offer a competitive program for graduate students, they have innovation grants as well. Also with our Department of Agriculture, through some of our malting and barley type, malting barley research. And always the disclaimers that the SCAB initiative require for anything we might touch on with that. So pretty broad base with what we do. I've had other funding in the past. I was funded by NSF and other capacities as well. So the basis for this, this was actually while I was still at the University of Wisconsin. I taught a course on data and exploratory and graphical methods. And I started to think about this topic as I moved back to the US, looking at my notes and thinking about the evolution of this data-driven world over the last decade. And what we do, more accessible coding, the incorporation of larger data sets. You know, one of the challenges I had in Costa Rica was accessibility. From a computational side, I had students, you know, I was teaching all the statistics courses in the college there. I had students come up saying they could not go to their home, have good connectivity to do anything outside of the classroom. So the models we had to flip around. So all of these things were kind of filtering there as we move into this digital revolution. But one of the things with big data, there's still a lot of unknowns in the agricultural side. Concept of big data for us is very different than if I'm in the human epidemiology world in many respects. And we can see that with some of the things we're wrestling with, we're dealing with in terms of how do we generate the right data to ask and answer some other questions. So our approach is trying to model strategies or scenarios to improve the understanding of sustainable crop production and management. You know, this is a baseline example we kind of use. If we target the right pathogen with the right product, we're making an assumption there that we know the pathogen is present and that it'll be activated in the loose sense, i.e. infection occurs in that. But for a lot of our farmers, this leads to a gap in the connectivity between what they should do when they should do it and how we can explore some of that. And so that underlying principle is driven a little bit when I come back to what we're doing. Maybe it's not surprising, but when I came back to the US after a handful of years away, I came back to the exact same questions that I was receiving not that long prior, which made me ask, is it us or is it the question? And it's probably a little of both in some way, as we'll see. You know, and for us, one of the approaches is we want to extract out those tactical decisions, explore those, try to quantitate them to some way when they would be needed, but can we turn them into strategies? And can we use the computational tools over time to build a system for farmers to play with those strategies and explore that? That answer is still to be determined in some ways because the computational needs are pretty great to get there. But we're making progress on some of that as we build these sort of structural pieces in place. So how I've broken up the rest of the talk is really in three, what I call examples or stories. One that set the stage for what we're currently working on, or two projects we're currently working on for different reasons to build this. But when I came back to the US, some of you know Sean Conley, who's at the University of Wisconsin. I see smiles. The fact you're smiling could be good or bad. I don't know. I'll leave it at that. But they had finished some work under this benchmarking soybean production systems in the north central US. And running classification regression trees, just looking at kind of how things drop out. Foliar fungicides kept coming up as important. But they were worried they couldn't explain it. This is all observational. This is farmer data, farmer production practices. And yield at that field level in the absence of disease data. So that's a big caveat for today's talk. There's not a lot of disease data until the last part as we try to fill in the gaps. What you can see here is a fairly extensive database. Working through and doing the quality control, in this part, we were working from 2,738 spatially referenced fields. And about 3 out of every 10 we received a fungicide. You can kind of see that in map 10. So the task was, can we try to explain this to Wade? And how might we approach it? So for us in that work, and this is published and you can access that for more of the detail. Taking a random forest approach. So this is one of the key areas of machine learning that's often done. All of this is being done in our system because of comfort and experience, which also means as this field evolves, you're constantly checking and rechecking certain aspects of the code. So what we can see here are different cultural practices one might think about. And these are organized by what's termed the feature importance. So you take that factor out and you ask, well, how much of the variation exists because of the loss of that factor? So in order of priority, the planting date and where you are matters. And this starts to make sense. It starts to have a story. Soil conditions, pH, organic matter, all of these, what are you starting to work with? But if you look at some of this, and here's foliar fungicide, it has a strong signal in that you take it out, it has value. If you start to look further down at some of the other factors, you might see something we'll touch on a little later with regards to other things in the system and asking how relevant those are. But for now, this tells us at least there's a signal, but is that signal uniform at protecting yield or at having an improvement? So from this, we can build the model and run that model to test that sort of condition. And what we do here, these are Shapley values and they're quantitative metric when you isolate out different factors to tell you how much above or below it. And in this case, it is the single field predicted mean minus the mean predicted yield, which is around 3.8 kilograms, or tons, that should be, my apologies, or 57 bushels. If you look at that, so we can start to break this out and we quantify the role of using the fungicide in that equation. So these have an interpretable value. So if you think about anything above 0 here, that means it had a positive influence in protecting some aspect of yield. A negative value, we're going this way. But you'll notice here the Y scale is very narrow. We're talking about 0 to 200 kilograms. So in bushels, we're just talking about a few bushels at best. In this example, we isolate out the 100 highest and 100 lowest yielding programs. So we're looking across a large spatial domain. You can see here unsprayed in this light blue. The orange is this color is the sprayed field. The squares are the lowest yielding and the triangles are highest yielding. So we have separation, which is what we expect. We're focused on the tails here to try to maximize it. What's interesting is we protect the yield with the spray, and not a surprise. But when I look here at those fields that did not receive a spray and have yield conditions that are similar, you don't see the converse. You don't see a negative. And actually the highest yielding environment was a non-sprayed field. So again, it's not a one size fits all. Maybe a little different in the lower end of the tail, some flipping here, but again, you're starting from a yield potential that's at a very different place. So it's a different question altogether. And we're probably in the plus or minus 100 kilogram range at this point. As we are able to drill down in a database like this, these are the top end, these are the 90th percentile, so just the top end fields to try to figure out spatially where we can explain some of this. What you see here are stratifications by maturity group, because we have that information from the farmers, the sowing date or planting date, and the heat units, the growing degree day accumulation. So a lot of these we estimate from models to do that sort of work in the same capacity. So I'm just isolating this out. Again, what we start to see is it's driven by a specific location. And for the most part, we're talking here about pockets of Iowa and Illinois. So not a surprise again, based on what we might expect with yield potential. But the key part is the earlier you go, this is April planting, mostly. So pushing up the planting date, driving the heat units. We have other data that we can explore. We know it gets us to flowering earlier with the current varieties, which extends the grain fill period, but it also extends the period of risk we might expect. So our ability to extract that using these models in this example is quite good. That's all well and good, but if we don't incorporate the economics in this sort of system, and this is a low input cost, so a low end cost to the fungicide in a high grain price scenario at the time, we're still missing part of that story. And so here in those situations, so again, the most favorable from the farmer's perspective, these TEDs are called technology extrapolation domains. They come from the physiological modeling side based on drought conditions, so soil aspect in some of these sort of scenarios. We can see the positive benefit in those. So it's telling us, the power of these sort of tools is telling us we are able to extract some of this signal out of there and explain it away. Now to do this, though, we have sitting there a very basic document that's 500 pages long to look at individual pieces. Because ironically, when this paper was submitted, somebody said, why don't you just run a bunch of t-tests? And luckily we were able to extract that information and say, yes, but now we add value to some of that. So what this result really told us, outside of really specific areas, the far northern range of soybean production tiers, the economic benefit was seen when prices were near or above that average. So there's a simple benchmark. And right now we're seeing prices that are below that point. So the farmers ask the sort of question again about this and trying to understand it. Yield potential environment's the significant factor though. Where you are and when you plant is really valuable here. But more importantly, not spraying doesn't translate into a loss. And from a disease side, what I've seen in our soybean systems with things like frog eye leaf spots or Cosperos vagina, we have had no impact for the last 5 years. And so this is telling me I can translate that data around and know I'm not going to lose something by not spraying in that. One of the key points we've come up with here is this non-universal profitability. Probably accounts for the discrepancies when you're talking to industry or whomever else it may be observed across these trials. Why might it turn positive one time, negative the other time? So again, the ability to explore some of this, we are at least able to provide context for what a general sort of, let's just throw the data against the wall and see what sticks as it drills down, comes around for us. But that wasn't all. Coming back to the original sort of model and rethinking of it, at the same time, the adoption of seed treatments in these sort of systems increased dramatically. And I've got some data to back it up, but if you kind of look maybe more recent terms in the last few years, we're talking easily 90% adoption, if not more, of using it. And that's a combination of fungicides, insecticides, and nematicides coming on that seed. And this one is really hard to get a handle on, both from an empirical approach, as well as you'll see with the modeling approach. How do we drive the model to try to ask and answer a question about that? So when we pull it apart a little differently, because we were curious, we just said fungicide, maybe that's not telling the whole story. This just illustrates it doesn't matter which component is in there. We don't see shifts in that respect. The good thing here is some of these shifts are minor in that we might flip latitude and planting date, but they're still at the top. And we actually don't drop foliar fungicide out of the equation. So we're not giving up something in the broader sense of the scenario. So we kind of look at this and we ask a question. And for us, we had to go back a decade to some degree in thinking about this. This was empirically derived data where we could show economically under certain scenarios that, and this was based on Wisconsin data, early to third week plantings in May, given the environment. But if the grain price was low, there was low probability of covering the cost in these low yielding environments. And we could see it was a function of the grain price. So again, some signals in the empirical trial, but is that telling us the story when we look at it from a farmer's perspective? One of the economic challenges with the seedling disease complex also as we look at it, and I pull out Phytophthora here, and we'll come back to that as something that we'll see at the end. if you look at the losses, it's a downward trend since 2018. Overall, planting conditions drive it. For us, if you ask the farmer what's the number one issue at planting, it's slump. The rest of it, they could care less about, to be honest. So these losses here are also questionable from the economic side of this thing. So what might be some of these drivers? And we don't have the resolution with the data to look at it, but we have ways to try to ask questions to go forward on some of it. A good example, I was at a meeting only a month ago, and Channel Seed, which has got a big foothold in the Northeast, which is owned by Bayer, they were already trying to line up contracts for seed for next year in early September. So trying to get at this has implications in the broad sense. One of the things we do know is that there's a lot of uncertainty what's on the label and what farmers understand. This is work I did with colleagues who were with the Economic Research Service and in entomology, trying to look at the data availability, which has its own set of issues in this respect, but trying to understand what's going on and what do farmers understand about it. One of the things we start with, these are 3 different labels for soybean. With farmers, I often say, can you tell me what's on the seed? There's one example where it's fairly clear. There is one where it is interesting, whatever standard means in that. The only thing I know is allevel on there, so I can pull that piece out. And then this is Roundup Ready 1 technology off patent. And you have to look very closely to see it's actually untreated in that example. So again, when we ask farmers this sort of question, many of them, they rip that tag off, it got put in a notebook, and then it got lost somewhere down the road, or it got washed in their jeans at some point. And this is a challenge because you have to do a lot of digging to understand some of that. And a lot of this, we actually have questions where it may or it may not fit in the system. The data available also to understand how these numbers change have been very different. So this is the share of acres planted with treated seed. Tech, which ran this for USDA NAS for many years, dropped the program because it was not cost effective to go out to figure out how many, and it was really based on sales of seed, not actual use. ARMS, the NAS work has done sort of different surveys at different times for different crops, but overall we see that trajectory in soybean in particular remain high. Corn is even higher a little bit in that, and so we get numbers that are all over the board. The irony is in our system in the Northeast, the one crop that probably does need the seed treatment is wheat. For a winter wheat with our planting conditions and our scab conditions because of the rotations and other factors and the wetness in the fall. And so kind of this contradiction of what we know from the biology and what is actually being done at times. We also know from other work, and that involve colleagues here, Christian Krupke, among others, when we look at empirical data, this is focusing on the neonicotinoid. We know that issue comes up, but the foliar fungus, or the seed treatment fungicide, also comes into that conversation as well. So we come back to our basics and we say, okay, what kind of question can we ask and answer in this sort of situation? The approach we took on the foliar fungicide as we matured the modeling approach, we started to question that approach. It's not really getting at the question we need to ask and answer for this situation. We actually need to know, if you put the treatment on there, what is the causal relationship? I would have to run a lot of trials to do that under a lot of conditions. Can we work from the farmer data to do some of this? So the baseline model, which follows from what we had previously done. So this is the same sort of profile here, just with kilograms. And it's mainly because we were less than 50 kilograms pulling it off the model we developed. We felt like this wasn't addressing the issue in the proper context. We really are trying to explain a causal relationship. If you do it, you get this back. So while the algorithm we had in the initial example does well for predicting the system, we're looking at it a little different. If you need some light reading, and I say that with a smile on my face, I would recommend Judea Pearl's work. There's some interesting examples about, you know, if the guard only shoots to kill the prisoner because they got the order, and then all these scenarios change, all these counterfactual, kind of makes you think for a little bit, but this is the approach we needed to take. We can interpret some things on a causal basis, but it's really conditional on some assumptions. So we're trying to separate that out. We have to change our mindset a little bit in the modeling approach, which you'll see. So what does a causal model look like? Everybody's favorite example for a plant pathology class is the disease triangle. Host, pathogen, environment. Everything comes together, you get disease. Everybody should know that's a really simple assumption. There's a lot more caveats to it. One of the challenges with this sort of approach, here's an example anthropogenic influences where we try to look at how the key drivers may influence either the pathogen or the host. So weather fits into both of those. And you can see some other sort of interactions. Think of this as a structural equation model. If you're familiar with that sort of approach, it has some hierarchical basis for it. We have a simple model here just from an illustrative purpose for looking at virus vector effect. The challenge here is all these models could be wrong. So we usually start by throwing a model up on the wall and arguing about it. Did you think about this, or which direction should the arrows kind of align? But they have some structural basis. The key here is what we are working from is the idea is we see the end result. So in our case, we see yield, and we're trying to build backwards the argument on it. So for us, can we quantify the amount of yield provided by that seed treatment based on the farmer data in this assumption? There are some advantages if the data sets are out there. We amplify our number of observations very quickly, the geography and the production factor, so we can cut across this. This, at the end, I'll mention, you know, if you're thinking about how to think about climate change, this may be one approach because we have areas that probably align to some climate change factors, but we'll have to generate the data sources to do some of that. The way the data are collected, at least in our example, in our situation, it allows us to extract a positive negative question with treatment, without treatment. We are relying here on previous research. Here's one example about farmer statements and reliability. The data are trustworthy. In this system, they have no reason to lie to us because they're freely providing us the information. And we work with them that way. So, you know, if it's stuck out as an outlier, it should be caught in some of our tools. The one big disadvantage is it leads to heavily unbalanced data sets. And so you may not always have spatial structure to the data across the geographies we look at, depending where you are. So what we can see is high yield variation. This just illustrates it. Now, because of the way we're asking the question, our quality control, we get to work from a larger database because we didn't have the same structure with the foliar fungicide. So we actually have a slightly larger observational database to start from, but you can see here across here, we're at 71%, which aligns to some of the estimates at the same time. And you can see the makeup across the geography. And, you know, for us, there's enough comparable locations of both types. So the approach here is a little different. We have a prediction model for yield, but we also have what's termed the propensity score, which is the isolation of that factor of interest. So this is almost like a testable, this is a testable hypothesis for us. We insert that in, we more or less look at different model structures to estimate and quantify its effect on that system. One of the big differences that from our first example is for us to make sure that it's not an artifact of something in the modeling structure, we actually took 11 different estimation approaches, and within each of those are different caveats. So at the bottom here, you see these line up. Differences in means would be a very simple, I did it and I didn't do it. What's the difference? That would be your t-test mindset. This is also probably what most farmers are seeing when they talk to industry representatives in many ways. So we take a series of approaches from causal forest, random forest world, Bayesian approaches, the G computation non-parametric approaches with the data as well, ordering it. Here, it's very hard to estimate heterogeneity in the system without doing a large bootstrap sort of approach. So all of these are the confidence intervals around the average treatment effect. And what you see is some consistency across these different modeling types coming out, but they're all very different from a very basic. I did it and I didn't do it. What were the two means? They're all lower and they all come in at approximately 63 kilograms. So not quite a bushel, if you think about it in the system. The range was anywhere from 3 to 189 kilograms in that respect. And this is only the fungicide seed treatment part in that. But it allows us to also compare this to what is known out there. Now we have something to play with in that modeling. We actually tried to force the models to use the fungicide seed treatment to see if we could extract the same spatial resolution we did on the foliar fungicide. But we couldn't actually identify any subgroups that would clearly explain why. So it's a rather random process and a lot of factors probably more involved than we're even aware of, or it's just random noise at that point. One of the things we did do, and this is the last part, this is actually, it was only completed a couple of weeks ago because we knew we needed to put a linchpin piece to this sort of work. So we put it in economic terms. And so these are an average soybean price over the last 2 decades. So we built a scenario around the model. And you can see the peaks and valleys. You can see 2 large peaks that held up pretty well outside of a few things, but here's the situation we're in right now. So it allowed us to do some sectioning out of the data to do the similar sort of exercise where we have the frequency of response. So we generate the data under a model. We use average seeding rate and other production factors to start this scenario. And the only thing we're really changing are the cost structures at the end of the day. We selected the model from the Bayesian additive regression tree, the SMART model. If I go back here to show you that, that's up here. Kind of shows a little bit of variation. It's somewhere in between the estimates to some degree. It's a little hard to see, I know, on the screen. But what this tells you, and the way to read this is the fungicide seed treatment cost dollars per unit. This is low, medium, high cost. The frequency of the observation, soybean price, this would be a range for lower price, say 6 to 9, 9 to 12, 12 to 15, and higher as the structure, getting across those ranges. And this is the net benefit where 0 sits in the middle and positive, negative. So when we are in situations where the seed treatment cost is low, medium, or high, but the soybean price is low, we typically don't see that as a positive benefit. It's a loss to the farmer. In that mid-range price, it holds similarly. So this is important as we see fluctuation in prices as we look at the overall potential across all these locations. And it's not until we get on the top end prices do we start to see the separation out. So it is driven by some of the economics, but overall the response is rather low to the environment. And there are other more important factors we would look at in this sort of situation. How do we translate those sort of results? I come back to the Phytophthora example and what it can do. And the reason for that is what we ended up doing to try to further explain it away is we just went to the empirical databases. What we knew was out there. The larger graph here is the frequency in terms of percentage response. We just did a simple yield ratio fungicide seed treatment against no fungicide seed treatment in those empirical trials. And so the ratio here tells you something about an impact. And if you look here, almost everything is around 1, so 1-to-1 ratio, no really difference. And in all of the examples, outside of a few with low stand issues or heavy rains within the first 3 weeks, our field's heavily infested with Phytophthora soja. The empirical work is nice. It allowed somebody like me to say, I bet all of them came from Northeast Ohio. And they all coincided with Andorance's research. And so allowing us to try to understand the combination of those factors is really critical in that. Our inability to predict it based on these models is validated in some ways by the lack of clarity across the empirical trial. This has importance because it goes back to understanding what's in your system to try to drive some of those sort of issues as they come forward. But it also tells us we have a gap in the data structure. So the last part I'm going to focus on for the story is how we're bringing it back to the field. Everything we did is in the absence of the actual disease observation. We can build a model, we can ask questions about that, but we also have to do a lot of data mining in that structure. So over the last 3 years, under a data-driven solution for soybean production, we have created the Open Crop Manager tool and been testing it across multiple states. With the idea of one, improving quality control on the data as we look to open this up to a broader research community and making this more accessible to everyone, as well as having a data protection and data privacy set of rules in place for the farmers to understand exactly what they get out of it. It's allowing us to fill in some of the gaps and either explain what we've started to see in the models or build off of those models to start to do additional sort of research on some of these type of causal relationships. It's a large team across UW-Madison and Penn State, and within Penn State, Anna here, one of my colleagues, she's actually an expert in data security, data privacy. We were internally funded for aspects of it, and then a large collaborator profile across the North Central region to do testing to kind of build up these tools in that respect. Our big picture vision is how to document the practices a little better and add layers that we didn't have in the original database structure. This also requires a lot of training to do it. AI digital imagery can work in the long run, but we still need to know what's in the field. And we thought, let's go this approach to add that layer as we go forward to do some of this work. The type of data collected is very similar to what we've been doing, but the big difference beyond is we have spatial resolution at the field scale. So we are in the field doing assessments in a zigzag pattern, or depending on the situation, we may look at it a little differently in clear areas. Ear damage would be one example of where you probably want to spatially define that more clearly. We have a similar production survey. This can be done at the end of a growing season, entered in about 5 minutes if all the information is available. Ideally, we want yield data at a map layer. We are also learning that is harder than it looks to secure in a lot of parts. But the other valuable piece is temporal data, the scouting reports that are done with some frequency. The structure to this and why it's valuable with our modeling approach is we define the manager being the farmer, who then can assign somebody to run the operation, a crop scout, an extension educator, you name it. And then within that, through our training, we train crop scouts. So undergraduate students to go out, do the scouting. They have different degrees of accessibility to the same information. The scout's going to see the field and the scouting information. The manager, co-manager structure is a little different. And we put the field at the center. The main thing, and working with our colleagues, the farmer or other users in a similar capacity are the data owners. They have the right to say you can use it or not use the information, which means as we develop the sharing mechanism off of this, you will see the same layer of information available. And the user, i.e. the farmer, has the right to retain that ownership of all data, and they can pull it at any point they so choose. For us, the farmers supported the project. It allows us to build the tool. They can use it how they see fit in many respects. So in our initial testing up until this past year, and we were updating information recently, we generated in 2 years of testing about 7,800 scouting reports. in this area. And you can see one example of how that might look over the entire growing season, going at different locations in the field where you can see what was assessed at those times. We can generate for the farmer a report back to it. The other nice feature wasn't a primary aim with our study, but we had the capability to capture and save images. And these are iPad, phone, you name it. They're not drone imagery or anything like that. We have the capabilities and other ways to explore that. So we have a range of things from abiotic stressors to diseases, insect pests and others, as well as weed images. And so that's something that we've had some conversation how to put that out there in different capacities across that. Why is this valuable? As I said, the techniques we can explore in the modeling do as well, but if I want to understand why something may or may not work, this is providing a layer I don't have. So this is the timing of disease severity in Pennsylvania from 1847 report for our major targets, white mold, SDS, and things like that. And what you can see is one that should align to what we know biologically. So that's a validation on the scouts. And it also helps us to explain a little bit better. If I highlight here, frog eye leaf spot, if you look, the severity is all less than 20%. So the scale here is 1 to 10, kind of 10% increment. And this continues to match up what we've been seeing. So it's a good validation. On the insect side, I showed this to John Tucker, our entomologist, and he smiled. Well, not because of white-tailed deer, but that's a different issue, actually, we're dealing with, nor groundhogs here in that respect, our two favorite friends. But when he sees this using the same structure, the severity here is going to equate to defoliation in that sort of system. Again, it remains at a low level, and it reiterates what he trains people on. Yes, insects are out there. Identify them properly and understand that they don't actually get to the damaging levels in many of the situations we do. And so this is helping us fill in the gaps we can't do in some of those modeling reports. But we are able to build off of that because now it's giving us a layer here. This is actually an example work of one of my postdocs, Santosh Angel, to build the same sort of causal relationships, isolate the planting date factor out, because this sets us up for going back into the databases to say, how much are you giving up when you miss the windows, and how does that influence the downstream? And so trying to integrate those two layers together is the direction we're heading forward on. On top of this, one of the things we also recognize in a system like this is the complexity of going across fields of different sizes and shapes. I sit in Pennsylvania. We have a lot of 5-acre, 10-acre fields. We can get across a lot of fields fairly efficiently, but if I go all the way across the Corn Belt into the Dakotas, and I'm talking 2,000 acres, it is a very different beast. And so we have started to build a layer across this, and we're using Sentinel-2 imagery in this situation to do this. Can we start to extract using NDVI as a simple measure to identify stress that we can then relate or correlate and make our sampling efficiency improve? One of the other things this is also leading us on is this generative AI and how to build models and then use those to generate the data. Because in spite of the data we've been able to secure to build this piece, it's still probably not enough to do the classic separate out data for model development and model validation. So can we build the models, generate the data, and then use the existing data structure? This maybe has a little bit more relevance in other systems, Fusarium head scab, where it's taken 30 years to amass 1,000 observations across a large geographical area. But we can build a model, but we need other sources. There's other things with the images we're looking on. And one of the last things I'll mention when I talk about the deer damage, actually Penn State has this NITNI AI Alliance. So these are undergraduate students in engineering, computational or other. And they work on projects like this. So for them, they are getting experience trying to understand, can we predict deer damage? the simple answer is probably no. Because the signal, but at least for them, this is a challenge. This is all funded by Lockheed Martin. Because for them, it's the value of the experience working on a real, on a practical issue. On top of it, they've had some experiences because 3 have never set foot in an agricultural field, 2 have never seen soil. So luckily or unluckily, my wife, who's the educator in the area, spent 2 hours with them, not only identifying what they were looking at, but all the other things they would have to think about. So they're getting kind of a shotgun approach to some of the things we deal with on a day. So different avenues for us, and we're always looking for options here as we explore the data sharing structure in that respect. So I think that is the last slide I have. I think there's been a little bit of time for questions. I'm going to stop here and I'd be glad to take any questions anybody has. So thank you again for the invitation.
Questions from the public. Maybe if we can start with students. No, look at our questions out there. Are we pushing the button here to ask the question? So yes, if you're asking a question, please push the button. Hold the button and ask your question. Faculty? If we start here. So what I guess that I'm gonna be talking based on our experience. And so data analysis using machine learning. One of the biggest challenges, I guess, that we are having is not only the resolution of the data, but also the reliability sometimes, and the amount of the data sets, because sometimes these models are hungry for data. And so, as you mentioned towards the end, sometimes we only count with just a few data points in some cases. But so what is your perspective maybe for the future as people start to use these type of approaches?
No, that's a fair question. That's actually a really good question. So at least from a pathological perspective, we have very few large scale data sets. Like I said, I use the Fusarium head scan example as one, and that's coming from a series of coordinated trials and lots of observations over time. So there's been a key investment for that part. But even then, what we amass is really only 30 to 50 new observations each year. But I think the power, it comes back if you look at some of the mechanistic modeling approaches that are being done. That allows us, as long as we know and you don't need a lot of validation data, you just need the right validation data, which is got its own set of issues. But if you have the right model to construct it, you can, through some of the more modern tools, can start to generate that heterogeneity into the system where you hope you can validate the data set you do have. So instead of trying to build a model off of a few observations and then every couple of years reevaluate the model, use that, hold that data back, use a different modeling approach, mechanistic or otherwise, to generate the data structure. But you need that's probably a published model, or that's you developing that model yourself. And how you parameterize those models are typically either based on empirical results or what you can mine from the literature to start to structure it. But it gives you a foot in the right direction to mass data that we can't do fast enough at a field scale. And this is why you see oftentimes the approach is to do coordinated type trials, take a meta-analytical approach because it gives some resolution to space and time. But beyond that, how do we take that even to the next step? It's one of probably several perspectives you can explore.
I have a question from online from Dr. Escalante. How feasible is it to continue updating the information to generate the models that counteract changes through time? For example, climate change or changes in management practices?
No, actually, we're wrestling with that on a grant proposal. right now, and we've identified a couple model systems we can explore. Our argument there through some of the generative modeling approaches where we generate the data is that if the resolution of our existing data set already captures heterogeneity in the system, so I'm thinking here climate change for that example. If it gets across the heterogeneity, we can actually input some of the scenario aspects, what may change for temperature of that. And our hypothesis is within our existing database, we have an example that illustrates that purpose. So that's one approach we're trying to deal with. We actually have to lock down the proof of concept on it. And our model systems for that are again, either fusarium head scab or white mold of soybean, because there are existing models that will at least allow us to construct that.
Paul. So I know you're still going in this direction with the sentinel imagery and stuff, but have you found a good way to differentiate or a good model to start piecing out what is stressing the plant, right? Is it a disease? Is it a weed? Is it herbicide injury? Is it something else? Or are you viewing that as this gives us a 30,000 foot view? Now we know a hot spot, we're going to target it with other approaches from there.
Well, so if anybody's looking for a project, so I think what we can do from the last 3 years of testing, because we have some spatial signals here, we're typically across 10 points, they're imperfect, but we can actually trace backwards to work with the imagery now and see how that overlays with that based on the signals, because we're capturing there, it could be abiotic or various biotic factors. So we may have a way to look at that. But if you look at those two test images, they're really aligning to what's most likely on the periphery of the field. And so in that case, I would have to go back to the image itself to see is that tree line, that might for us get back to the deer damage sort of question based on the type of data we're seeing. So how do we use some of that to at least explore that? I think we can do it, but it's a step up again in computational aspects. Just to generate those test cases is a fairly substantial computer run. And that kind of goes back a little bit to Christian's question. These are computation intensive. For those working with drone imagery or anything like that, if you're using RTK technology, like we were talking about, Tommy, you are amassing a huge amount of data at that field scale, and you just have to have a computer, whether it's a supercomputer or a really fast mini supercomputer, just dedicated to running that data for you. It's the sort of thing I learned in statistics from my advisor with scenarios and simulation analyses. That's the sort of thing you go to the gym, you go home, have dinner, you go to bed, you come back, you have breakfast, and it's probably still running kind of world we're dealing with. So, and how we store all of this. This is another thing. Most of us at university with our systems, we have the capabilities to do some of this, but you just imagine all the people working, whether it's microbiomes or other data intensive sort of pieces. It's going to be a big issue as we go forward.
Maybe a follow-up question, shared to what Tom said, and so multiple groups around the world are working on these different issues, different crops. And so something interesting that you mentioned, based on your data, not spraying deep, not translated as a logs. I'm assuming that you refer to foliar fungicides.
In the first example,
Right.
yes.
So then, what is your perspective of the future? So if we compare the current ways, how we do things, decision making and all that, and based on the evidence that we have found, and maybe what is coming, using these methods, or what do you think might change in the future and how?
Well, I think it all feeds into a larger system. For us, this data allows us to build different questions that we can answer different scenarios. So in an ideal sense, it would be the farmer sits down and plays like my video game is old school, I guess. It's probably more Pong or whatever it was called. But they're allowed to change those parameters to explore, to build something local and then try to keep that resistant to change. The example I can show you on the foliar fungicide with the fluoride spot in Pennsylvania. We are running with that data to tell them not to spray. We have seen it consistently because we don't want to be in the mess that the Midwest deals with. And the Mid-Atlantic, Mid-South deals with, because in the push to understand this, all this, that pathogen is overcoming it, resistance is occurring. We're trying to have a data-driven solution. Going forward, can we mount it? I think I was talking with John Wallace, because he ran a trial for the spot spray technology with some of the equipment. It amasses a tremendous amount of information, but as he said, I have high resolution on 400 acres, but I don't have spatial resolution because we couldn't get it into other fields. We have a lot of rich, and then just trying to sift through this. I think for farmers, that's always the catch-all. The farmers, I don't think that's going to change as we amass more and more. Most farmers, by the time they can sit down with maps and other information, at this point, they've already made the decision. And so we have to try to build this a little more dynamically for them to sit down with it. It's not quite answering your question, but it's how we have to think going forward and modify that. So the tools exist that allows us to do updates, and as we open this up, I think anybody who has a question hopefully can feed it back in, whether it's a model, prediction model can help drive some aspect. But we need kind of a team effort on some of this. No one group's going to be able to solve that problem.
Right. And so you have experience in the tropics as well. Bananas, Costa Rica, et cetera. So you know how intensive it is. So I think the conversations out there are, if we can reduce even these number of sprays by 10%, that's a gain, right? So do you think that would be possible based on these data intensive models?
Well, there's some advantages there because the system and mainly in the scouting data, there are data that exists already to explore some of this. The challenge is that like in Costa Rica, they switched the model to all drone sprays. And all the conversation I've had here in the lab today and all the conversation we continue to have, that has its own set of issues right now on the quality. So there, my concern is they're just flying, the drones are just whizzing by all the time, but they have no idea what the width is, what they're spraying, what the impact is, what the pathogen is doing to respond to partial sprays, not getting a good coverage, because there's no way to easily see that. There, for those who know the banana industry in Costa Rica, that's 42,000 hectares of pure carpet banana. It's a long-term monoculture, so again, trying to extract some of that. That's always been the challenge. We can show you can PhD. We can show we can take out a spray right now and not impact the quality of the response. We can put a biological in there, so that is an option. But that's done in the absence of the tech side. That's just let's explore it based on the principles we know. So we can do it. Can we scale it up? That's going to be the bigger question. Yeah, there's.
So Paul, I'm talking to your growers then and showing this data, have they responded positively or what is it, if they changed? Or not, or?
I'm asking about impact.
I just.
I think the initial response more to the latter part, when we put data like this in their hands, they're asking more questions than they're used to. When we go out now and show them, and then we bring the real samples to the table, that has impact. Because they start to understand, oh, okay, just seeing it isn't enough. I have to go do something for it. We are starting to get questions, and I credit John Tucker in entomology because he's been, and Christian Krupke here, they've been beating the drum really strongly on the neonicotinoid. They are starting to get questions, where can I secure seed that isn't treated? So it's small victories, I would say, but we're seeing some positive response. But I mean, I was doing a program where I brought in a bunch of stuff, leaf spotting, corn, tar spot, all of this, and the farmer panicked. Like, did I screw up? And I'm like, no, because we had to show them it's really low. Less than 5% disease. Because to them, they just see damage. They see a symptom and they extrapolate beyond what they're really seeing.
Right, and so we're gonna run into that with the tar spot. Because they see it and then freak out in one county, but then they missed it in the other, and, you know.
And we have, this year, we have the same issue on that side because the areas where the risk would be considered greatest based on productivity and factors like that, the weather was not as favorable. It was very dry and the timely rains missed until late. But in the areas where you aren't able to ramp up, I think we were talking about that, you could see fields that were black by the end of the season. How does that translate going forward? In some of those situations, I think the farmers got to make some tough calls because these are also fields that have just, maybe there's something in the general aspects of the interaction, because these are fields that often also have white mold problems in soybean. There's something really favorable for the pathogens once they get. But can they do something about it? Do they want to go out there when they need to do that? That question's always.
Well, I was saying, on the frog eye side, we probably have no field changes and anything I've done since I started.
Yeah, I mean, I even have the industry out there and they actually, when they come out to check the plots in our situation, it's kind of humorous because they say, am I missing something? I said, no, you're seeing what I'm seeing. So are we in agreement? There's nothing here. And they go back and they inform their boss or their boss's boss, whoever they need to be. Does that change that? No, because when you get to the market, the marketing side changes that conversation. That's the hill we have to climb. So.